Hello, in this video we will understand about the introduction of insulin like growth factor 1 pathway and breast cancer progression due to BRCA1 mutation will lead to cause cancer. So in the previous video we was discussing about the insulin like growth factor 1 whole mechanism, how to interact and how to BRCA1 mutation. In this video we will understand about the general overview. Insulin like growth factor 1 is a potent mitogen of major importance in mammary gland. IGF-1 binding to the cognate receptor IGF-1 receptor. Insulin like growth factor 1 receptor triggering a signaling cascade of phosphorylation lead to proliferative and anti-apoptotic event will lead to cause cancer. Experimental findings have demonstrated that normal rat mammary epithelial cells continue to proliferate in serum-free media in response to insulin-like growth factor 1 in the in vitro growth, suggesting that the insulin-like growth factor 1 system play an important role in mammary gland function and maintenance and growth of the mammary gland. That is why insulin-like growth factor 1 is important for the mammary gland development. This is the female contain mammary gland contain lobe. This lobe is internal structure is the this is the mammary gland. An internal structure of the lobe is the lactiferous duct are present, which that is open into the nipple for the lactation of milk, and the lobule will be formed inside of the lobe. Extralobular duct are present here, and this is the total terminal duct lobular unit. This is the lobular unit terminal duct. Let's find the further acinus. So in this way, the lobule contain acinus, many acinus contain lumen inside, which that is used for the milk storage. Let's begin to understand the acinus wall. This acinar wall inside is contain epithelial cell, while also the macrophage, dendritic cell, and the CD8 T cells are present for the pathogen combating and inhibit of the infection due to the innate and adaptive immune system are present. This orange color line shows the basement membrane, while this is the orange color cells show the stromal cell. This is the fibroblast, is used for the collagen and connective tissue formation, contain the FGF receptor, the fibroblast growth factor receptor. This myoepithelial cell are present used for the constriction will lead to ejection of milk due to the oxytocin production. Stromal extracellular matrix as well as the here is the adipocyte are present. So the stromal cell are present in the extracellular matrix. Stromal extracellular matrix. So the BCRA1 mutation will lead to cause upregulation of the insulin-like growth factor 1. Remember, primary stimulation, let's begin to understand that when growth hormone will increase, will lead to primary stimulation, and the growth hormone will bind with the receptor, growth hormone receptor will lead to cascade of phosphorylation to trigger the chromosome 12 with the DNA gene activation to produce insulin-like growth factor 1 as well as insulin-like growth factor receptor. Insulin-like growth factor receptor and insulin-like growth factor 1 will produce and autocrinally bind itself with the epithelial or stromal cell, while the epithelial or other also paracrinally or endocrinally mean the insulin-like growth factor when enter into the blood circulatory system will target the other tissue to proliferate further. Let's quick overview about the breast tumor. When the epithelial cell will trigger the insulin-like growth factor more, will lead to proliferate and differentiate. In this way, the original cell is the different, while the proliferative cell will be different, and this is going on the stage of the metastasis. Insulin-like growth factor will bind autocrinally and paracrinally. Also bind with the stromal cell will lead to enhance further insulin-like growth factor 1 major production from the stromal cell. So the stromal cell paracrinally will release the insulin-like growth factor will bind with another host tissue cell. In this way, the paracrinally and its cell will produce more insulin-like growth factor 1 will lead to differentiation of the cell, as well as the differentiation will lead to metastasis, and the tumor formation will proliferate and it disperse throughout the body. Let's begin to understand and find the mechanism. This is the stromal cell or breast or epithelial cell contain a insulin-like growth factor receptor. Phytomumab is major inhibitor for this tyrosine kinase inhibitor will inhibit the binding with the IGF-1. MAP kinase and AKT will stimulate when bind the insulin-like growth factor will lead to overexpression of the SP1 gene, while the inhibit the BRCA1 gene and P53 and WT1. While the survival cell due to the lysosome and proteasome inhibition due to the cascade of phosphorylation, AKT and MAP kinase. So in this way, the SP1 gene will overexpress because inhibition will impossible due to the MAP kinase will inhibit this gene to inhibit the BRCA1 major gene will lead to not regulate the IGF-1 receptor. So the uncontrolled transcription to translate the IGF-1 and IGF-1 receptor will lead to target another cell and proliferate the uncontrolled and survive the damaged cell even will lead to cause uncontrolled proliferation and division cause cancer in the breast, in the mammary gland of the female. So the estrogen is the major source which that trigger the SP1 gene. When estrogen will trigger the SP1 gene when bind with the estrogen binding protein receptor or estrogen binding protein will lead to trigger as a transcriptional factor to produce more IGF-1 receptor and IGF-1.